Hi everyone, it's Misato again. Hope you enjoyed DNA Part 1. Now we're going to talk about RNA and DNA. So today's aim is how does RNA help DNA keep the cell alive? These notes we're going to learn about replication and transcription. DNA has two jobs. Job 1 is replication. It's the process by which DNA copies itself to DNA. So think about it when I make photocopies of your notes and I keep making a copy. I still have the original, but every time I make copy, I'm making a copy of the original. Job number 2 is protein synthesis, and that's the process by which the cell makes proteins. If you guys remember, when we talked about cell organelles, ribosomes makes protein, so think of rib. Ribs are considered protein. So the ribosomes interprets the message from DNA about which proteins to make. Job number 1, replication. DNA makes more DNA. So the replication occurs when chromosomes copy themselves before cell division. The diagram below on slide number 4 shows an example of how replication occurs. We kind of call it unzipping, meaning that the DNA strand is unzipping and a new strand is being made on both sides of it. The new strand you'll see is the black, and that's just showing you one DNA strand being replicated into two identical DNA strands. This is an example of replication. You have the original strand and you have the new strand. Now, I know some of you are going to ask the question, well, what happens if there's a mistake? If there's a mistake, we usually call those mutations. Sometimes mutations can be good, and sometimes mutations can be bad. Some mutations can cause cancer, which is an uncontrollable cell growth, or other mutations can cause you to maybe have 2 different color eyes. If you guys have seen huskies are pretty cool dogs. They have different color eyes, and that is a mutation. Sometimes a mutation can be that you have freckles. Doesn't mean it's bad. Sometimes it can make you different. This is another diagram of DNA unzipping and a new strands being made. The second job of DNA is what we call protein synthesis. And what's the goal? Well, the goal is to make a chain of proteins outside the nucleus. Why? That's because proteins code for different traits. So if you remember in our DNA part 1, I said that your DNA codes for proteins and those proteins code for different traits. So there's 2 steps in protein synthesis. One is transcription and the other is translation. You need RNA for protein synthesis. RNA is a little different from DNA. RNA just happens to be a copy of DNA that has the ability to move into the cytoplasm and actually leave the nucleus. And that's pretty cool. And RNA stands for ribonucleic acid. There are some differences between DNA and RNA. RNA is single-stranded. Uracil binds to adenine, so there's a new base. There's no more thymine. So for RNA, A is going to bind to U. Your ribose is going to replace your deoxyribose sugar. That's another difference. So there's 3 differences. It has 1 strand. Uracil is a base instead of thymine. And the sugar is different. It's ribose instead of deoxyribose. Why does DNA need RNA? Well, DNA never leaves the nucleus. It's the master copy of all the directions a cell needs to stay alive. So it needs to be protected. That's like if I locked myself out of my classroom, I know where to find the master key. But if I don't have that master key, then I can't get into my classroom. So that's why DNA is so important. You can always make more RNA, so it's okay if it gets destroyed, but you can't make more DNA. So I gave you 2 kind of diagrams down below just to remember that DNA in the nucleus is safe and DNA in the cytoplasm can be destroyed. I hope you guys were able to follow along. Please don't forget to fill in the blanks in your note sheet. So sometimes the blanks could be the yellow words that were in the PowerPoint. I will also post a plain PowerPoint with no voice recording. I miss everybody. I hope you are staying safe and healthy, and I hope to see everybody soon.